Hello everyone, welcome to my channel Online Biology Tutorials. In this video we will study antibody and its various subtypes. This is the core topic of the human health and disease chapter, and this is ultimately important. Stay tuned till the last, every bit of it will be discussed. So let's start with our topic, antibody. Antibody are the proteins that are forming a major part of our immune system. They are also known as immunoglobulins. Okay, these are also known as immunoglobulins, and they are abbreviated for this as capital I, small g. They form the major part of our plasma. What is plasma now? Plasma is the liquid component of the human blood, and these antibodies, they form 20% of the plasma proteins. Antibodies, they are always a part of acquired immunity. Whenever we talk about humoral immunity, basically we talk about antibody production. So today we will see how actually an antibody looks. What are the different parts in an antibody structure? So we will start with our structure making of the antibody. So this is how our antibody heavy chain look like, right? Let me tell you one thing. If I talk about any of the antibody, then there are two heavy chains in one antibody and there are two light chains. As it is a protein molecule, that means it's a polymer, and the monomer unit for it would be amino acids. So there are 440 amino acids in the heavy chain, and there are 220 amino acids in the light chain. Whenever we write any polypeptide, we write it from N to C terminal. So if this is our heavy chain, then this is our N to C terminal. You can see that there is one heavy chain here and the second heavy chain here. Now we will make quickly the light chain. Light chain, as it contains less number of amino acids, then of course it will be shorter than the heavy chain. Okay, so these black structures, these are our small or the light chain of the antibody. Whenever we want to show antibody, we can also write AB as well. This AB is an abbreviation for antibody, and this AG is the abbreviation for antigen. So here also we will show the N to C terminal of the light chain. Now one more thing, the terminal part of both the heavy and the light chain is known as the variable region. Okay, V here means, yes, you are absolutely right, V here means the variable region, whereas the region beyond it, it is constant in nature, means the number of amino acids are fixed. They cannot shuffle. That's why this region is known as C for constant region. Now this region is the fragment for antigen binding. Okay, fragment for antigen binding. That's why it is abbreviated as capital F, small a, small b, Fab region. You can see these structures in the between of the heavy and the light chain. They are basically the antigen binding site. Means this will bind to the antigen. And antibodies are very much specific in nature. Each antibody has a specificity for the antigen, means it can bind to specific antigen. Okay, so similarly, this region here on the left, this is again FAB, and it also contains antigen binding site, which is abbreviated as ABS. So we do have ABS here, we do have ABS here. Now the region just after this FAB, it is known as FC. So the region after this, right, from here, this one, so all the yellow colored parts are your FC, fragment of crystallization. Okay, so let me repeat once again. There are two heavy chains, there are two light chains. Heavy chain, why are we calling it as heavy? Because its molecular weight is more, because it contains more amino acids, and the number of amino acids are approximately 440. Whereas the light chain is light because it contains less number of amino acids and its density will be definitely less. The terminal parts of the heavy and the light chain, they are variable in nature because the amino acids may change. This is just to have ability to bind to different types of antigen. Okay, so terminal region is sensitive, hypersensitive in nature, it's variable in nature, and in the terminal region only antigen binding site is there. The terminal variable region is known as fragment for antigen binding, which is abbreviated as FAB. Whereas the region beyond it, it is known as FC, or the fragment of crystallization. Right? Another thing which I have to tell you is that disulfide bonds are present between the heavy and the light chain, and for connecting two heavy chains together. So here you can see at the fragment of crystalline region, I am showing S-S. These S-S means the disulfide bonds, and these are the strongest covalent bonds. Disulfide bonds are formed when amino acids, they have sulfur in them, like if methionine is there, cysteine is there, then if two amino acids will come close, then disulfide bonds will be formed. So here we have shown in the case of heavy chain, you can see that they are connecting heavy chains together. Now, similar type of bond is present between the heavy and the light chain as well. So you can see I am connecting heavy chain with the light chain, and these are our again disulfide bonds. So they will be present along both the sides. Whenever you will make it, please do make a little more gap than I have done. Okay. So disulfide bonds, they help in connecting the heavy chains together. They also help in connecting the heavy and the light chains together. So this is the general structure of an antibody. Now various variations are being seen in their structure, because of which various subtypes of antibodies are seen. So our next heading is going to be the subtypes of antibody. Now all the antibodies, they will exhibit the basic structure that we have just done with. Now antibodies, as I said, they are abbreviated as IG. I here means the immuno and G refers to the globulin. Okay, so it could be IgM, IgA, IgD, IgE and IgG. Remember, antibody, it is always abbreviated as capital I, small g, and the name of the subtypes, which are M, A, D, E, G, they are to be written always with a capital letter. Now we will discuss one by one their characteristics. And IgM is the biggest in size, okay? So it's pentamer in nature, means the structure that we discussed above, this will be repeated 5 times, okay? So let's make its structure. This is our antibody 1. This is our antibody 2, okay? This is our antibody 3. This is our antibody 4. You can see that I am showing heavy chain and light chain. So accordingly, you should also know. Now you can see that 5 antibody structures are being made, and this black line actually shows the J chain. This is a short chain that helps in connecting one monomer with another. When I say one monomer, that means I'm talking about 2 heavy chains and 2 light chains. So this kind of antibody, it is pentamer in nature, and as you have seen that each antibody has 2 antigen binding sites. So this is our 1 antigen binding site, this is our 2 antigen binding site. Because it is pentamer in nature, that means 5 antibody structures we have to make. So if 1 has 2, then how many antigen binding sites will be there in 5? Yes, there will be 10 antigen binding sites in the antibody IgM. Okay, this is the one antibody. Whenever any infection occurs, this is the one which is released at very first time. This is released at the first go. Okay, so IgM is a pentamer. It has 10 antigen binding sites, 1 monomer of antibodies connected to the other with the help of J chain. Right? Now let's come on to the next one, which is IgA. And IgA, it is also known as secretory antibody because it is present in the milk of the lactating mother, and it is also there in the colostrum. When a mother comes in the lactation phase, the first milk that is ejected from the mammary glands, it has a color of yellow and it is rich in the antibodies. Okay, so the antibodies that are there in the colostrum, they are IgA, and they are also present in the milk that comes after the secretion of colostrum. Okay, so IgA is also known as secretory antibody. Where is it present? It is present in the milk and the colostrum. It's a dimer. When I say it's a dimer, that means 2 monomers of antibodies are to be drawn and they will be connected to each other with the help of J chain. IgD is less in our body, and this is one which is related to the allergic reactions. Right? It is the main antibody which is related to the allergy is IgE. So I am writing here only the main antibody related to allergy is IgE, but IgD also helps in this process. Now 75% antibodies in our body, they are the maternal antibodies that we have derived from our mother. So IgG is the one which we have in our body that are being transferred to our body with the help of mama's blood. So this is the one which has crossed the placenta and it is there in major amount in our body. Okay, so all the 5 subtypes of antibodies are very important. Their names: IgM, IgA, IgD, IgE, IgG. Maximally we have IgG in our body. IgM is a pentamer with maximum antigen binding sites, and it generally binds to antigen that have polysaccharides on them, because each antibody has its specificity. So if I talk about IgM has a specificity for those antigens that contain polysaccharides in them. Polysaccharide means many glucose units, many saccharide units are there. IgA is secretory in nature, it's a dimer. IgE is the main antibody which is connected with the allergic responses, and IgD is less in the body and it helps IgE. I hope this was helpful. Stay tuned to my channel for more videos, and if you haven't subscribed till now, please click the red button beneath the video. Thank you for now. Happy learning.